Dr. Manish Kumar is engineering from the University of Tokyo, Japan. He joined IIT Gandhinagar in 2017. He has more than 20 years of research and teaching experience. He worked at the University of Nebraska, Lincoln, USA, Kunsan National University, South Korea, Uppsala University, Sweden, and Jawaharlal Nehru University, New Delhi, India. He have contributed more than 150 international journal articles.
So thank you very much, I convener and co-convener of this program, who has invited the Dr. Goyal and Dr. Jyoti for giving me this opportunity to share my research in terms of a kind of text or class lecture. So I had to modify my general talk, that where we think that we start with a result and like that. But here I have been told that it has to be a classroom lecture kind of thing. So today I am going to talk about wastewater-based epidemiology as a surveillance tool for wastewater monitoring. So let's start with what is here. Maybe some of the term are new, maybe epidemiology, surveillance, and those things can be the new for some people. And of course, for me also, epidemiology was not I was not much aware of this until lately. So this is, so let me explain first what is epidemiology. These are some of the viruses that we have made, which in the context of this we will talk. Sometimes I forget in the end to acknowledge, so that's why I always bring it first, to acknowledge my collaborators and funding agency. So what is wastewater-based epidemiology, WBE? So as we can see that the epidemiology is derived from the three Greek roots, that is epi, demos, and ology. Ology is, of course, the to study, and demos is the people, and upon. So a study upon the people. So a study of the demography. So if anything that you don't need to, so then epidemiology refers to a population, a distribution of a population, and we can determine several things, the things like biological, social, economic, political, administrative, and legal thing, that the control of health problems, and by determining the population's psychology, chemistry, biology, pathophysiology. So these things we can determine of a population or a demography in a given location, or the distributed in a given catchment or even given state or country. So that is, in mere sense, it is epidemiology. So we can say that epidemiology is a study of population, how they behave. And of course, the population, so we have to find, we can determine that any property of a population that is distributed among themselves. So that can be a virus, that can be a disease, that can be like the epidemiology of tuberculosis or cancer epidemiology. Even the problem of teeth can be a part of the epidemiology. But so the scope is very wide. It can help us to the management and investigation, not only that where and what, but also by knowing that you can manage it. So originally it was started, epidemiology was invented for managing the epidemics. But now it is being used for any kind of communicable, non-communicable disease. And now, recently, it can be applied to all disease, all health kind of problem, even not only the health issues, but several other things also. So here, with another term that is being used is epidemics. So I would talk about a little bit what is the difference and what is the epidemic. But before that, what are the type of epidemiology? So the type of epidemiology in general is two types, descriptive and analytical. We can define frequency. So descriptive is descriptive, right? That a kind of qualitative information that we may derive from this study that can be in form of case report or case series. It can be based on individual or population, and so the ecological, the cross-sectional, cohort, prospective, retrospective. So there are several types of descriptive analysis can be done, which essentially will tell us that how much frequent a particular event or particular properties are there, what is their distribution. So basically answers four major questions: how many, who, where, and when. Let me take the later pointer. So this is the type of epidemiology. So I already told you that what is the origin, and so it is a and what types of type and scope. So even if to summarize it, we can say that epidemiology, the importance of epidemiology is that it allows development of guidelines for the prevention and control of certain disease. So by studying that, you can say mask pehniye, ya 6 gaj ki duri, and so and so forth can be, many guidelines can be implemented to control it or to manage it. And discipline that make up epidemiology. If you are doing epidemiology, you have to be knowing about the statistics. You can say that, okay, I want to do graduation in epidemiology. Of course, we have a lot of epidemiologists, but this is a kind of, you have to have a basic degree, and then after that, you can do a specialization in it, where you have to, you will be taught, or you will have to know ecology, microbiology, sociology, statistics, and psychology, and many more, maybe RT-PCR, in this case of COVID-19 surveillance, or even RNA extraction or wastewater quality. So depending on that, what you are going to do, or what is your purpose of epidemiology, which disease you are targeting, the many other specialization, many other information keep on being added. So this is the how epidemiology we can talk about. Then before we move to the wastewater surveillance, it is very pertinent to share that what is the difference between epidemic and pandemic. But before epidemic and pandemic, which is a little bit easier to know that, okay, the epidemic is a concentrated one and the pandemic is widespread. But what is the difference between epidemic and endemic? So endemic is a regular found disease among a particular people or in a certain area. Like some disease are only in the certain part of Africa, or some disease is only in certain part of India, or certain ethnic group because of their culture, or so and so forth. So those kind of disease is known as endemic, like malaria, tuberculosis, chickenpox. These were termed in endemic. But epidemic is a widespread occurrence of an infection disease in a community at a particular time. So which is, so at a given time in a community, it can be widespread. So which means that we can say that North India got an epidemic, or Kerala got the Ebola, or the SARS, and so forth. And rate of infection continuously increases. But in endemic, rate of infection does not increase, it remains constant. So it's not that today it is 5, tomorrow it will become 50, but epidemic, it will increase. So and the epidemic is characterized by spreading, but endemic is localized with a certain person, certain ethnic group, and certain geographical location, and rate of infection remains constant. So the epidemic is, example is a smallpox, H1N1, and black plague. Now, this is a very nice diagram that I found from the internet, that Epidemic is you look that a certain location area, a certain part of the globe has certain disease which has the constant infection. If it is being reported 3% of the population, it will remain even in a decade 3% of the population. But and so like that. So I'm sorry that the cursor has to be here. So in this is the endemic and certain part of. But the epidemic is that it will grow. So with the time it will spread. It may be the epidemic. Yeah. So an increase in the number of the cases, and the pandemic is where you can see that several parts of the globe at one time with the increasing infection rate. So then when in the endemic becomes global, we can call it pandemic. And yeah, so this is the difference between pandemic and epidemic. I have 45 minutes, so I talked about what is epidemiology, what is epidemic, and now I am going to explain you what is wastewater surveillance. So the wastewater surveillance is that there is a, like, let's suppose clinical surveillance. Clinical surveillance that there is a population, you get one person came to your clinic and then you tested and you said that whether that particular person is positive or negative. Wastewater surveillance is that if such a community, you don't need to take the sample from these people, but the wastewater they excrete, that wastewater you can sample, and we can sample and say that whether how much percentage of the population seems to be affected or at all there is any infection in this group or not. So this is a community-based the surveillance or epidemiology. So epidemiology is the study of the population, which means either you go and talk to person, or you can take some indicator that can give the health of the community. Like many times, sale of a medicine. You don't need to go to the home, but just nearby the clinical, the medicine shops, you can know that what is the frequency of particular drug being sold. And you can know that, okay, what is the case. Polio eradication, also wastewater surveillance was used, whether the polio is eradicating in a certain group or not. Bill Gates, the foundation, or the Melinda Gates Foundation, is that they had this polio eradication. There also the wastewater surveillance was used to see whether polio virus is still coming to the wastewater or not. So essentially, this diagram is from one paper, and which was formed for COVID-19, wastewater-based epidemiology, global collaborative to maximize contribution in the fight against COVID-19. So here you can see that, yeah. So this paper so was made last year in February. And I am one of the authors. So here, essentially what is there is that this is a magnifying glass, right? So this is a surveillance tool, I mean representative one, and it is looking at the wastewater and finding the various, and then of course, if there is any outbreak in a population, it will have the fecal setting, it will come to the wastewater, and in the wastewater you can understand that what is happening, and you can give a response that how to manage it. So the wastewater surveillance can help you to control something in the population without going directly to the population, because wastewater characteristic or quality of the wastewater is very much dependent on the population fingerprinting. It says that the lifestyle, their drugs use, many things they can be, it will be reflected by the wastewater. So the problem of a statement and gap identification, this is part 2. So the part 1 of introduction part I have finished, where I have talked about epidemiology, then the difference between pandemic, epidemic, and endemic, and then we talked about what is wastewater surveillance. Now the problem of statement and gap identification. So when we talk about the wastewater, it's not many places there may not be the wastewater treatment plant. However, still this is a published paper in Hazardous Material Letters of ours, where we have predicted that, look, in the hospital there will be virus, of course, COVID-19, and the buildings and like that. And if it will go to the urban or sewer system, and there will be some leaking from here. There may be rain, there can be combined sewer overflow. And then it can go to the wastewater treatment plant, and the wastewater treatment plant can treat it or may not treat it, but of course you can find it in the wastewater treatment plant. And because these viruses are so small and nanomaterial, nanoparticles, nano-sized particle, it may get rid of the drinking treatment, the steps, and it may or it may reach to our drinking water. Burial of the infected people can also lead to the groundwater contamination. If there is a wastewater treatment, the dumping site, that can also leaching and septic tank and those things can bring the virus in the river, lakes, reservoir, and the aquifer system. So we talked about this. So it means, and we don't know any coefficient that how much really or not. So this all is a problem or gap of the information that we will come to the COVID-19 the surveillance, but let's talk about the wastewater surveillance. So wastewater surveillance is not a new thing. This paper is in 2018, where the researcher tried to find the early warning of hepatitis A and norovirus outbreak. Norovirus is a gastrointestinal virus, and if you get infected from this, you will have the upset stomach, diarrhea, basically. So what they did is that they detected the genome of the virus in the wastewater, and this was the red dot where the observations, and they put the line diagram. Nearby health center, they collected the number of patients complaining or this kind of symptom, norovirus symptoms, or diagnosed with the norovirus. And they plotted this, and it is they connected it with the black lines. What we can see is that the patient peak in the hospital has arrived after 2 weeks of the infection peak predicted through wastewater-based epidemiology. So this advantage of the 2 weeks is a big advantage. We cannot lessen it. And similarly, it's not that we were waiting for COVID-19 and then only we did the research. This is our previous work that is published in 2019, and we sampled it in 2017. This was occurrence of antibiotic resistance, viruses, pharmaceutical care products, PPCPs in ambient water of Guwahati. So this yellow shading is urban drains. This is not, the in between is the river, R1, R2, R3, and this is lake. And we have determined the PMO of norovirus 2, IIT virus. So these things we have determined, and here it is so we have done it much before, and you can see that the comparison. You can read this paper or find. So PMO is paper mold virus, which is only the virus that is being excreted by human beings, because this virus is found in chili and no animal eats chili, or so that is the virus we used. And this the blue line or red line is a seasonal variation in that. This is pre-monsoon is the red one, and the blue one is the monsoon. You can see that norovirus was not detected in the monsoon. So this is in anywhere. So this is, but in pre-monsoon, it was detected significantly in the river, lake, and urban drains everywhere. So it means the dilution or the rainfall, everything comes into the play. Now coming to the COVID-19, the wastewater surveillance of COVID-19, let me quickly introduce you what is COVID-19. And this COVID-19 is a disease which is caused by zoonotic virus. And zoonotic virus is nothing but a virus which is transformed into the secondary source. So a virus which was originally in the primary source could have eaten by the human and no problem. But if it goes to the another, if that primary source is eaten by the secondary source or reservoir, and that virus get changed or mutated, and that mutation cause or enable them to cause the disease to the human being. So the bird flu virus cannot directly infect human virus, but H1N1 virus, which is a modified bird flu virus, can, and that is a genetic virus. Similarly, the COVID-19, it happened that the bat is the primary reservoir, and which get modified by pangolin, and somebody ate pangolin, and it's infected the human being. Any virus disease is measured based on the x-axis contagion, how much contagious it is, and how much deadly it is, y-axis. And which means that cholera, which is much more contagious, but not much deadly. Bird flu is very much deadly, but not so contagious. But if we plot COVID-19, is neither too much contagious nor too much deadly. And maybe that is why it becomes pandemic, because even an infected person for 10 days or 7 days do not aware that that they got infected from the COVID-19, and they act as a carrier and they spread the virus. But having said that, SARS-CoV-2 is not a new virus. It is a seventh member of Corona family virus. Corona family, a family which is named after their crown-like structure or envelope surrounding them. And it has different viruses, even including SARS-CoV-1. And COVID-19 is, of course, CO for corona, V for virus, D for disease, and the 2019 is a year. And SARS-CoV-2 is, we always know that virus is something between living being and non-living thing. So then, but among the viruses, there can be double-stranded RNAs, double-stranded DNA, single-stranded RNA. So this is the tiniest possible the entity that is single-stranded RNA, which is a span of 60 to 140 nanometer. So when we started, we tried to know that what is this virus is all about. So we wrote this paper, a chronicle of SARS-CoV-2 epidemiology. Epidemiology. So we tried to understand what is the epidemiology of this, diagnosis, prognosis, transmission, and treatment. And this paper we wrote, and this diagram is from this paper, where we tried to say that a virus should have been originally in an animal place, and but it interfered the human life, and then it is going to be. I already talked about this and how they are interlinked. But here I would rather emphasize that even wastewater surveillance of COVID is not new. I mean, the SARS-CoV is not new, because in 2012 and 13, Kyle Beebe, who is a well-known virologist, he took the many sample from 10 sewage sample from 5 wastewater in the US and scanned for all DNA viruses and RNA viruses. So DNA virus like herpes virus, adenovirus, TTP virus, parovirus, and RNA virus like coronavirus. You can see that they found 83% of the sample positive with coronavirus. And then they concluded that abundance of newly emerging viruses like coronavirus, the strong representation of the respiratory virus, and relatively minor abundance of occurrence of are increasing. So they already said that respiratory viruses are getting more and more abundant in the wastewater treatment plant. At the same time, in 2020, this paper has claimed that SARS-CoV-2 infection leads to its persistent fecal shedding from the excretion point of view. So which means that if one infected person is likely to shed the virus, and that virus is likely to reach a wastewater treatment plant, and if we detect that, we can find. We can know that if there is a in given community there is some problem or not. Wastewater-based epidemiology, global collaboration, collaborate. So, and then this was the testing is not enough, and in India very high much. This one I already talked, and so we made it for COVID-19. Then this paper came from the Australia. This is the first ever paper that detected SARS-CoV-2 gene in the wastewater. So this is also little bit a kind of methodology that virus excreted from a person through the toilet and the hospital, they reaches to the wastewater treatment plant, and so there is a virus. This virus was RT-PCR, and RT-PCR is polymerization of chain reaction. So it means a tiny particle of the gene can be amplified and found that how much gene is there present in the effluent water, and this effluent water is now calculate SARS-CoV-2 per capita load. How much genes is in the wastewater, now divide it by the population, and you will get a per capita load, and then compare with the public health data, and then we can know that whether there is a matching, and you can forecast, and you can use this information for good epidemiology management. So this Ahmad et al, they did in the Queensland and the different type of, but what is very clear here is that not only determining the virus in the wastewater, for which you may need to be a to run the RT-PCR, you may need to be a virologist or a biotechnologist. For understanding the wastewater, you may need to be a environmental engineer. And then for epidemiologist, again, to understand the population and the statistic and those things, and per capita load, you may need another skills. So it needs a different kind of thing, but here they already have shown that, okay, the different colors they have provided and matched with the population density, and they try to correlate with their study. Although this is I mean, I would say the preliminary studies, because the few days data has been shown, and with the less loading, because in Australia, the number of patients were not so high. So what is the methodology? How to do that? I already told you that you came to know that surveillance, you can see that this is a surveillance. So a CCTV, as soon as we hear the word surveillance, we remember this surveillance camera, which is watching us. And so in this case, we can watch the virus, and this is a wastewater, and we can know that, okay, how many viral genome per day is coming, and with that we can compare with the surveillance or the population wise. Because and this wastewater surveillance is much useful, because clinical testing or the observed case might be just the visible part of an iceberg, because there may be in the population where this disease has lot of asymptomatic cases, many cases might be hidden, might remain hidden. So again, getting back to, we need to understand the structure of this SARS-CoV-2 virus, and this is a spike protein. See, this spike protein has the S gene. So we, this is a envelope, and then there is a capsid and RNA and N protein. So this is a sequence that is typical for SARS-CoV-2. So we cannot determine only one, because maybe the sequence is unique, but the and that makes or separate it from SARS-CoV-1 to SARS-CoV-2. So here what we have done is that we calculated the three genes, ORF gene, I mean the measured or detected S gene and N gene. What was the procedure is that bring the sample, put it in the centrifuge tube, centrifuge it because, and take the supernatant so that any gross particles, sediments, soil, they will be precipitated, and then you filter it out from 0.22 micrometer. This size filtration shows that even the bacteria will be removed. So only what will be remain in the supernatant is enzyme and proteins. This, the centrifuge tube, we add the polymer, polyethylene glycol, that will just precipitation. So it will settle down, but when it will settle down, it will take viruses with themselves. And NaCl will make the charge favorable to us. This mixture of the supernatant after filtration and adding to the PEG and NaCl, we leave it for overnight. And overnight at 100 ppm, it will shake little, and then precipitation will occur. After that, we will take some sample in the microliter, 2 ml. From this 50 ml, we can take 2 ml. From 25 ml, and then we will do the ultracentrifugation. Ultracentrifugation is important to not let even a single virus be lost by the supernatant. So this such a high centrifuge so that all the virus is settled down. So a pellet will be formed at the bottom, and whatever the left water is there, that water we can throw out. And then we start extracting the RNA. So there is the RNA extraction kit, we can do that one. And then once RNA is extracted, we start to doing the RT-PCR, reverse transcriptase, the PCR. And this PCR will give you this kind of result. You see, I told you that three things that we measured. We measured N gene, we measured S gene, we measured ORF1AB, and this red one is the control. So we add some known virus concentration, MS2 phase, in the sample, so that if I am not sure that this water has virus or not, I cannot check whether my analysis is correct or not. So what we do is that after all this step, we keep on, we add a known MS2 concentration so that we know that this much virus of this type we have added. So if our sample analysis does not show that, it means our analysis is wrong. So this is a control, positive control. So you can see that the green color VIC is a N gene. So this is a green color. So this is the N gene. FAM is blue. So this is a ORF1 gene. So this is ORF1, and this is a CT value. So what happens that in the first cycle, if there is a 1 gene, in second cycle, it will be 2 to the power 2. It means 1 gene becomes, so 2 to the power 1, it means 2 gene, 2 gene will become 4 gene, 4 gene will become 16, 2 to the 8 will become 16, 32, 64, and so on and so forth. So 2 to the power until 40 million of the amplification occurs. So even if tiny particle is there, at certain number of the cycle, it starts appearing. So MS2 phase, it has appeared at here around 20 CT value, but these three guys appeared around 30. So that is, it means their concentration is less than the MS2 phase, but they all are prominent after a certain CT cycle. So if CT value is higher, the gene concentration will be lower. So what we did, of course, this one, but there is another method. So actually we did in PEG precipitation because at that time supply was not good. But the many places they wanted to use ultrafiltration or ultracentrifugation process. Not all have done what gene I detected. They like see the CDC, N1, N2, E gene. Somebody has only E subraco. So it depends on what kind of kit you are using for RT-PCR. So who is the supplier? And so it keep on changing for that. But PEG method was not initially was not so popular because they think that it is more precise is ultracentrifugation, but it needs a lot of labor because ultracentrifugation is like from 1,000 liter to 1 liter. We have 2,000 times enrich the sample and then check that whether there is a virus or not. But we thought that okay, the PCR, the PEG should be there. So what? But before that, so we did an experiment of both two, and we published that work that where we compared the two techniques. So and we found that polyethylene glycol method had superior inhibition removal than filtration method. So filtration method is not only taking the time, but it is also producing a lot of error. If even one drop of filtration has not happened, or we removed the filter and so on and so forth. But apart from that, because wastewater has a lot of unnecessary enzyme and proteins which they compete with the virus, because at the end of the day, RNA is also a nucleoprotein, right? So in that case, we found that polyethylene glycol is a much better technique than infiltration, and we published this this was our second publication on this topic. Precipitation of viral particles from the sewage is now being used. This is a snapshot from the paper, from the presentation, where they try to compare with the different. They also add MS2 phase, and they can do sequencing. And many times it can give you direct the gene number and like that. But the most importantly, what have been achieved in this process is that they can know the even variant. So this is a wastewater treatment plant, and so the detected mutations. So they can know that okay, what kind of mutations are occurring, which kind of the code or gene is no more prevalent. So wastewater surveillance can also help you to detect the mutations. So of course, many people have drawn it at different ways. The earlier that I have shown 250 sample, centrifuge, filtration, addition of PEG, then again overnight shaking and all that. So same thing. And this, but here one more step is that uh, this data can go to a data mining or a certain Excel file. And if the in charge or primary investigator sees that data is fine, they can send to the portal, and we can use or inform the government that particular area may have some problem. I have a little bit, I have to be hurried, but this in China, when I was reviewing the paper after publishing these two, we found that the one case was that before treatment, they didn't detect SARS-CoV, but after treatment, they detected. That has opened my, I mean, how could it happen that a treated water, which is coming, is not having the gene, but the influenza, but the treated water has. So here, what I did is that we take the sample at every step: primary treatment, then USB inlet, USB after aerator, aeration tank, and clarifier, and we have seen that what is going on, how the virus is decreasing, or if any such observation is there or not. This one was published in Science of Total Environment about the decay along the treatment plant. So one thing we also made it clear that it's not important that it will remove everything, but major part of the world were showing the treatment plant can remove the gene. So far, this is a conceptual depiction and findings. So far, this is how it works: aeration tank, effluent, everything I have shown that okay, that this is a virus and centrifuge, and then those things are RT-PCR. reverse transcription, DNA, RNA, cDNA, and amplification curve. So this is a simple depiction of everything that I have shown. But now let me discuss some findings and then we can conclude. So this is the first finding, that in the first time we wrote, our group was that first proof of the capability of wastewater surveillance for COVID-19 in India through detection of genetic material. So this was the first ever study that we reported. And here we have shown that 8th May or 27th May, how with the 8th May, there was a less gene detected, but less COVID patient was there, and 27th May, patient number goes high, and wastewater also shown that detection. After that, what we did is that we did some capacity building. We tried to help, and in the due to the KTH was the common person, so we could help Bangladesh to detect the first ever in their wastewater treatment. They didn't have the wastewater treatment, so from their nala, from their drain, there was a detention site and in a stadium. So we calculated, we were successfully detected the SARS-CoV-2 in a canal and main sewer, and tried to know that whether the upstream to downstream what is happening, or whether canal the gene is going into the main sewer, so it gets diluted or it gets accumulated. In fact, we thought it will be diluted, but we had to report accumulation as per the data. So that was the interesting thing. Then the pandemic curve monitoring. So then that was only the beginning, and the war started there. Then we got the UNICEF money and also other funds from IIT, and we started this pandemic curve monitoring. And then we reported this 2-month data in September and August. We reported it in the environmental research, where we have shown that this genome concentration is red line and the patient number is blue line. And we showed that this peak of the genome concentration corresponds to this peak in the patient concentration, and so on and so forth. And always we get around 2 weeks of the advantage or heads on or in early warning. So we wrote unraveling of early warning capability of wastewater. Then there was a longer set of data, 7 August to 25th January, and this was a several things that we have tried to put and see that how patient number is matching with or not. And we were able to detect a surge, a spike along the valley, and also we were able to detect the second wave. But like I said, that my principal idea was that it is a nanomaterial, and so it cannot be removed completely, because at the end of the day, when RNA is fragmented, it is even sub-nanoparticle, and so it should not be removed. And so even if initially we could not find anything in the treated water, I let my team to measure. And so what we measured is that even now it is accepted in Chemical Engineering Journal. And we reported that, look, not completely the treatment or removal of the RNA happens. And we not measured only, we compared a conventional treatment plant and this is a root zone treatment plant, zero discharge. So here it is a root zone treatment and a conventional treatment. And then we compared these two and reported in the Chemical Engineering Journal, and now it is online. So that is the one of the things that is very unique, that we suspected, and our hypothesis was true, that it cannot. Then because this kind of work always remain in the media very highlighted, so they asked me some practical question, how to use it on the city scale. So for that, I did a systematic study that we divided the whole entire city into 6 parts, and we measured the SARS-CoV genetic concentration and compared it with the people number. And then we compared and we saw that it is working. So we published that as a city donation for effective COVID-19 pandemic preparedness powered by early warning. Then by the time we reach there, people start asking us that, well, the treatment plant have the RNA, so whether does it mean it will affect us? What is the transmission probability and like that? So we used some models and tried to give a ceiling of the potential risk that can have, which is not much, but still we cannot ignore it. And the percentage of N gene, ORF1 gene, and S genes were even we reported now it is under review that in the surface water, because if treatment plant is not removing all, that means that it is bound to be reflected or be present in the surface waters, in the ambient environmental water, and that what happened eventually. We also gave the scheme of called SWEEP, Surveillance of Wastewater for Early Epidemic Prediction SWEEP. This is a paper of ours in collaboration with IIT Roorkee. Here we gave this abbreviation SWEEP, so Surveillance of Wastewater for Early Epidemic Prediction. And SWEEP, where there we told that, okay, how to use it in domestic, in urban area, rural area, how to use the laboratory analysis data, whom to send first, and how to make the best out of it. And this is also available in environmental research. So the future implications, so this is what we have done so far, although I tried to go with very fast. But the future implications are that we have to be careful. We have to be careful during this COVID-19 management, because the medicine, handling the medicine is very much important, how much we take, because, and how much is left and how we dispose it, because we cannot digest or metabolize all, and it will reach to the, again, to the wild. And if such water with low concentration of such medicine will be drunk by the reservoir of the viruses, the virus in their gut will be immunized, will get a vaccine kind of shock, which will not kill them, but will make them strong. So antiviral resistance may develop in future. Then somebody also told that, how about a paper-based technique, which we dip like a pH paper, and it produces color. Similarly, it can give us that how much is the gene present in the wastewater, but it remain on the thinking scale. It could not, so far, not happen. I also, I had a data of antimicrobial resistance, anti-drug resistance. When a E. coli or bacteria does not listen to the medicine, that is called a resistance state. So in 2018, we had a data of Ahmedabad, some certain wastewater treatment plant or river and lake. Same thing we did in 2020. And when we compared, we found that it is increasing. Now this is available in hazardous material. Such kind of the papers and work draw United Nations Environmental Program attention towards my work. And I was invited for as an expert on a report that is being prepared, that is environmental impacts of antimicrobial resistance. And so wastewater-based epidemiology around the world. So this is what I did. This is what is happening in my research life. But it's not that what could have been done, or that was the max, because I saw that around the world, people have done much better, because this is a dashboard data flow. This dashboard, I am part of this, and this is like handled by that paper that I've shown you, global collaboration. What they do, that whatever the paper publishes on this wastewater surveillance of COVID-19, they put the data on the map and in this dashboard. So far they put 235 universities data, 59 and 50 countries, and more than around 1,500 sites are there. and they report it on the Twitter. They collect this system from the Twitter, Facebook, and other, and then they categorize and then they display. So they have used those that how to, even if somebody put anything with the COVID-19, they notice it. And how can it help is that it can help a planning of the city, it can help transportation, RT-PCR, graph mapping, several things it can be noted. How the world is adopting it is also a very much, it's not that it is reporting or somebody is reporting others' cases, but this is a nice Dutch Netherlands case where they, scientists does their part, some other people have put it on a GIS map, but the government also comes together, and then federal partnering has happened to develop a wastewater surveillance framework. And there are many agencies, EPA, DHS, NIH, USGS, NSF. There are many the stakeholders of non-government, community implementation. They all are coming together, and so at the same time, in science and technology evaluation for practice, community implementation, and testing search, all are occurring at one time. This is probably my last slide. So what COVID-19 sewage cannot do? It cannot tell you the number of people. I mean, if you have a certain gene number, you cannot say that, you can say per capita or something like that, but you cannot say that how many people must be infected there. Then the second point is that you cannot say, there can be false negative. False negative because of dilution. So if your wastewater sample may say there is nobody infected, but in fact there is some. But it can help you to detect the wastewater. It can give you a trend, whether the condition is bettering or it is worsening from the previous day. So negative results do not indicate absence of cases. Decentralized wastewater system will not be captured. So in the villages where wastewater is not being collected centrally, it will be not implementable, or it will be not give you the right result or desired result. But urban area where wastewater is going to a single place, we can use it. May be impacted by pre-treatment of sewage for odor or worker safety. So of course, pre-treatment and those aspects, or sewage collection, network system सही है कि नहीं, सभी पानी collect होता है कि नहीं. So those things can be critical. But apart from that, which other points are that, how important is the wastewater surveillance, whether it can do the early warning or surveillance, dealing with the variant, can we detect the variant? Just to let you know that we are also able to detect the variant now. So Indian variant, we have detected coefficient of comparison, that यहाँ के study का ऑस्ट्रेलिया के study से क्या comparison किया जा सकता है. No infectivity because RNA is not intact, and so some collaborations are needed. With this, thank you very much.